Hello, this is Mr. Philippek, and this video is going to be on cell division. Alright, so if you think about it, we start off as a cell. And this cell begins to divide inside mom into two, and then we keep dividing and keep dividing and keep dividing, eventually, till what? You're right, till it turns into us, right? Think about it. We started off as one cell, and now we're anywhere between 15 to 20 trillion different cells. And so we have to understand, how is this process where our cells divide? Now, scientists take a look and see the 4 different phases of mitosis. And mitosis basically is the name of cell division. Okay? And there are 4 phases. There's one called prophase. There's one called metaphase, Interphase, and Telephase. And a lot of times, we just like to write down the first 4 letters of each of those phases. We call it PMAT. All right. If you wanted to get fancy, we could just highlight the first 4 letters. Just a nice little mnemonic device here. PMAT, prophase, metaphase, anaphase, telophase. These are the 4 different phases that allow cells to divide. And so in the last video, we really spent a lot of time talking about this, which, if you remember, we called it interphase, otherwise known as the cell cycle, where the cell spends a majority of its time, where it grows, it develops, right before it kind of heads into this region here. Remember, we have to go through the duplication of organelles and all those other things. And so we're going to talk about the 4 phases here of mitosis. And so this is P, this is M, this is A, this is T. All right. And then this last little part over here basically is what we call cytokinesis, and we'll kind of get to that. And so the first phase, as you remember, P, is prophase. And a lot of neat things begin to happen here in prophase. And so, as the slide says here, it's the first and the longest phase of mitosis. And remember, in animal cells, we have these things called centrioles. And the centrioles, again, are right here. And these aren't found in plants, so they're only found in animals. They begin to move to opposite sides of the cell. And if you notice here, something begins to break down around the DNA, which is the nuclear envelope. And so all of a sudden, chromosomes begin to appear. And what happens then is that, as the chromosomes kind of pair up, because remember they had to get duplicated, we copied DNA and interphase, they begin to kind of match up. It's like a big square dance. Okay? Well, the next step is metaphase. And if you notice here that all the chromosomes have lined up, right? They're lined up across the middle. And the idea there is remember that each of these chromosomes, in order to get 2 equal cells, have to move in opposite directions. All right? So some chromosomes will head towards this centriole. Some chromosomes will break up and head towards this centriole. And the idea, remember, is we want to start with one cell and end up with 2 very identical cells, because that's how life has to keep going here. All right. Now, these little spindle fibers here are called microtubules. And these microtubules, which are right there, are the things that are going to grab the chromosome and kind of ratchet it back towards the pole, which is where the centrioles are here. So the next thing that has to happen once they all line up is, you got it, they have to split apart. And so these chromosomes are going to be pulled by those microtubules towards the polar end of each of those technically new cells here. Okay? Basically, it's going to keep pulling all of these chromosomes back in either direction until those chromosomes get right here. Or right here. All right? And so now we want to kind of guess what's going to happen next here. And that is, you guessed it, we get this pinching in of the cytoplasm. We start to see the formation of these two new cells. So if I take a little dotted line, I kind of draw around here. Here's one new cell. Here's another new cell. Okay. If you notice here that the nuclear envelope begins to reform, the chromosomes are going to begin to start breaking down. Okay. And so just to kind of flesh out some of these terms, telophase is this final phase. The nucleolus begins to reappear. And if you remember what the nucleolus is, it's that small little dot inside a cell nucleus where ribosomes are made. And so basically now what the cell is going to do is begin to pinch in and begin to form all the new parts of what the new cells are going to be. And so the final step in cell division is what we call cytokinesis. And this cytokinesis involves the pinching in of cytoplasm. Okay, and now we have the two new cells, and both of these cells are identical. And that's important for the continuity of life, because without it, you can't afford to have one skin cell to be different than the other. It would be interesting, I'm sure, but wouldn't be the best thing. Okay. Now in plants, something new forms here, and it'll get a little cute here. We get something called a cell plate, because remember in plants can ill afford to have a pinching in of cytoplasm. Sorry about that. And the idea is because remember plants have this rigid cell wall made out of cellulose, and so this cell plate forms right here and begins to form what is going to be the new cell wall. And eventually, after time, sugars, cellulose, begins to fill in where that cell plate is, much like in humans, where our skin starts off, or our bones start off as cartilage, and eventually develop into bone. All right. Same thing with plants. We go from, again, we'll go cute here, cell plate